Okay, welcome back for our last video on our look into the reproductive system. Last time we looked at the male reproductive system and the hormones involved, and that ended up being extremely simple. So let's look at something a little bit more challenging here, the female reproductive system and its hormones. So let's get into this. So when we were looking at the male reproductive system last time, our focus was on the testes. So it shouldn't be a surprise that our focus here is going to be on the rough anatomical equivalent in females, which are the ovaries. So if the main functions of the male reproductive system were the production, maturation, and storage of sperm, in addition to the production of testosterone, which is necessary to maintain those processes, those are all going to be necessary here in the female reproductive system, but with a couple of extra complications. Since the female reproductive system not only has to produce and mature the ova, the female gametes, it also has to be responsible for housing all of the developmental events that happen after fertilization. The fertilization to give you your zygote, and then its development into the embryo, and then everything up until the childbirth process. So naturally, since all of those extra events occur, the female reproductive system is going to have a few extra moving pieces, and the hormonal regulation is going to be a little bit different, just so we're aware here. Okay, so if we look histologically at the ovary, then we will see here that the actual oogenesis process, the actual gamete maturation process, takes place within the outer cortex layer of the ovary. So just like the kidneys and just like the adrenal glands, the ovary consists of an outer cortex layer and an inner medullar layer. So this cortex layer is where the actual oogenesis process is happening. So there's a lot happening in this picture, but if you actually kind of follow it from the top left and then you make your way clockwise, those are all different things happening chronologically speaking. So you start out with these smaller primary follicles, and then as they accumulate certain cells we will talk about later, they become secondary follicles, and then you get to the ejection of the oocyte in the ovulation process, and then you get into the latter stage of the luteal phase. So those are all things that we're going to talk about. So don't make the mistake of thinking all of these things are all happening at the same time. So this is kind of a time-lapse image of what an ovary might look like over time. So all of these things are happening in the outer cortex layer. That inner medulla layer is actually mostly just for infiltrating blood vessels, arteries coming in, veins going out, and then any lymph vessels to carry excess fluid away, and then any nerves needed for stimulation effects. So the equivalent of the spermatogenesis process in males is the oogenesis process in females. So there are some similarities here, but things are definitely going to be different. So in males, the germ cell, the diploid germ cell, was called the spermatogonium. Here it's called the oogonium. So everything's pretty similar so far. We still have a diploid germ cell, meaning it has 2 copies of every chromosome. Like in males, that oogonium is going to undergo a single round of mitosis to produce 2 primary oocytes that are also diploid. Now, at this point, the primary oocytes are then going to start the meiosis phase, which again consists of 1 and a second nuclear division, but these primary oocytes are going to arrest themselves in the middle of meiosis 1, meaning they're not even going to complete that first nuclear division. So those diploid cells will arrest here until the onset of puberty. So the production of those primary oocytes actually occurs much earlier in females than it does in males. Those primary oocytes actually start forming prior to the female being born. So these primary oocytes are going to be arrested here in meiosis 1 until the female child is born and until she hits puberty, in which case she will experience a surge of luteinizing hormone, which we also saw in males, but that was really just for getting the testosterone production going. In females, this surge of luteinizing hormone not only serves to get estrogen production going, but it also serves as the trigger for these arrested primary oocytes to go back into meiosis 1 until the point at which they resume it, and then they will eventually arrest again in the middle of meiosis 2 at the metaphase process. So we will talk about that a little bit later. Another major difference is that if you focus on spermatogenesis on the left side of this diagram, each one of the 4 spermatozoa that were produced from the division of every one spermatogonium germ cell, each one of those 4 haploid daughter cell products were actually viable sperm that could participate in fertilization. That's not quite the case with oogenesis. With oogenesis, you're only going to get 1 viable egg per division of an oogonium, so you get 4 times less opportunities here. So the reason for this is that every 28 days, which is the length of the ovulation cycle in females, that resumption of meiosis that we see is going to trigger what is called asymmetric cell division, meaning that every time those cells divide, you do not get an equivalent division of materials. Instead, one of what is going to become mature ova is going to get the majority of the cytoplasm and organelles and proteins and other resources, whereas the other potential eggs are going to be left with very little. So those other eggs are then going to wither away as what are called polar bodies. So the purpose of this is that because the egg, again, is going to, after fertilization, basically house the development of a whole new organism, a whole new human, you want that one egg to have as many resources as possible so that we can nurture and ensure the maturation and healthy development of the soon-to-be formed human child. So you don't want to take your chances and say, okay, we're just going to have eggs that all have the same amount of resources, because each one of those may not have enough. So you're basically putting all your eggs in one basket. Pardon the horrible pun, I am so sorry, but you're basically putting all of your eggs in one basket to make sure that one egg can succeed rather than having 4 eggs that none of them can succeed. So after this happens, this is where the now secondary oocyte is then going to become arrested in metaphase of meiosis 2. And it is then going to stay arrested there until the fertilization process, at which case it will then finish meiosis 2 and it is now ready to go. Okay, so something that is very interesting that I feel needs to be brought up. This is very interesting. So when fertilization occurs, you of course are producing a diploid zygote. So that zygote has 23 of dad's chromosomes, which came from the sperm, and 23 of mom's chromosomes, which came from the egg. So you put them together, you end up with 46 chromosomes, right? You have an equal amount of dad's DNA and an equal amount of mom's DNA. So it turns out that that is not the case for the mitochondrial DNA. Did you know mitochondria have their own genome? It's not very impressive when you compare it to the DNA inside the nucleus. The nuclear genome contains about 25,000 genes and about 3 billion base pairs worth of DNA. The mitochondrial genome is absolutely tiny by comparison. It only contains a little over a dozen genes, 13 or 14. So it's very, very, very small. But still, it is DNA that is capable of being inherited from person to person. So you might think, oh, well, it's the same as nuclear DNA. I've got half of it from mom and half of it from dad. Well, it turns out that during the fertilization process, the mitochondria that are in the midpiece of the sperm actually are not transferred into what will then become the zygote. The sperm actually only contributes its nuclear DNA, meaning that the fertilized zygote actually only contains mitochondria that were a part of the original egg. So the implication of this is that all of our mitochondria are inherited from our mothers. There's no such thing as having your dad's mitochondrial DNA. You always get your mitochondrial DNA from your mother. So I expect that once this lecture is over, you're going to call your sweet mother and you're going to thank her for her mitochondrial DNA, in addition to all the other things that she's done for you, right? Always good to show your gratitude towards your mother. Am I right? Of course I'm right. Okay, so let's get back to talking turkey here. So we are going to split up the female reproductive cycle into 3 different phases. The first is called folliculogenesis, the second is ovulation, and then the third is called the luteal phase. So we're going to talk about them one by one as they occur in chronological order, and we will talk about all the cells and the hormones that are involved. So again, if we look back on our discussion of the male reproductive system, the two major players here were called Leydig cells and Sertoli cells. The Leydig cells responded to luteinizing hormone and produced testosterone, and the Sertoli cells responded to follicle-stimulating hormone and helped us start the spermatogenesis process. We do not have perfect analogs here in the female reproductive system, but we come pretty close. Roughly speaking, the female equivalents of these cells are called theca cells and granulosa cells. So the theca cells aren't exactly like the Leydig cells. The granulosa cells are not exactly like the Sertoli cells, but they're pretty close. They're not that far off. So those granulosa cells are going to associate initially with our primary oocytes to form structures called primordial follicles. So the word primordial really suggests that we're just at the very beginning of this, which is appropriate. Luteinizing hormone and follicle-stimulating hormone, both of which come from the anterior pituitary, are both going to stimulate the growth and proliferation of the granulosa cells, so that is mainly a follicle-stimulating hormone effect, and the production of estradiol in both the granulosa cells and the theca cells. So that's a luteinizing hormone effect. So that's one of the departures from what we saw with the Leydig cells and the Sertoli cells. The Leydig cells were just testosterone production. The Sertoli cells were just spermatogenesis. Here, both the granulosa and theca cells are capable of producing estrogen in response to luteinizing hormone, but generally speaking, the granulosa cells are really what focuses on the oogenesis process. So this surge of both luteinizing hormone and follicle-stimulating hormone stimulates the production of the first primordial follicle, but all the synthesized estrogen that comes from the granulosa and theca cells is going to trigger the negative feedback loop that we're familiar with by now, and this is going to shut down any further follicle-stimulating hormone coming from the anterior pituitary, and it prevents us from making any more follicles. So only this first dominant follicle actually forms and survives folliculogenesis. Any other follicles that started to form are going to end up being too immature to really stick around, so they're going to kind of wither away. Only the first primordial follicle that was able to mature into the dominant follicle really sticks around to do anything, and this really helps us to explain why only one egg is produced per every ovulation cycle. Okay, so here in this picture you can actually see several different things that have already happened that we've discussed and things that are going to happen in the future. So after we've had a discussion on not only folliculogenesis but also ovulation and the luteal phase, you may want to come back and look at this figure here to make sense of some of the things that we only really got to talk about and we didn't actually look at pictures for. But one thing that we will go ahead and mention is that during the maturation process that we see in folliculogenesis, you are going to start to see the formation of that zona pellucida barrier around the oocyte itself. And as we said back in the male reproductive system, that serves as a barrier to anything that is not a sperm that has the acrosome that is capable of dissolving that. So it prevents anything from penetrating into the oocyte short of the sperm itself, which, as we said, is capable of dissolving the zona pellucida. Okay, so if we look one more time at the folliculogenesis phase here, we see that surge of GnRH and LH and FSH, which stimulates the production of estradiol in the ovaries, which helps to ensure that we only get that one dominant follicle forming. But in addition to that, the surge in estradiol actually will also target the uterus and begin preparing it for the possibility that we're going to be dealing with a pregnancy. And one of the major effects here is that the endometrium, the innermost layer of the uterus, the second one, the middle layer being the myometrium, which we've talked about before in childbirth, the endometrium thickens and prepares for the possibility of carrying a child. You want that endometrium to be nice and thick there. But as we also said, you do see the negative feedback with all the extra estradiol, which shuts down the FSH and LH and ensures that only one actual follicle gets made. So after the folliculogenesis phase, we go on to the ovulation phase. Well, I don't mean we as in including me, but you know what I mean. Okay, so Since there's only one dominant follicle remaining, that follicle, since it is made up primarily of the primary oocyte and then all the surrounding granulosa cells, which make estrogen, that dominant follicle is going to start making even more estrogen, and that is going to start a new type of feedback loop that we haven't really talked about very much. So the classical negative feedback says that once you make a certain amount of estrogen, that should shut down FSH and LH, right? That's classical negative feedback. But this follicle, this dominant follicle, is going to make such a large amount of estrogen that it's actually going to kind of confuse things a little bit, and it's going to trigger a positive feedback loop, which is totally normal here. It's not anything going haywire. This is how it's supposed to work. So basically we get a large amount of estradiol stimulating the anterior pituitary to make even more luteinizing hormone and follicle stimulating hormone, which of course triggers a positive feedback loop in which we get even more estrogen. So this surge in luteinizing hormone levels is actually going to trigger ovulation, in which the dominant follicle is released into the peritoneal cavity away from the cortex of the ovary. And the primary oocyte that is contained within will now start to resume meiosis 1. So the granulosa and theca cells that remain behind after that primary oocyte is ejected are going to stick around in the cortex of the ovary, and it is going to start forming a new endocrine structure called the corpus luteum. So this structure is maintained with help from the stimulation of a hormone that comes from the placenta called the human chorionic gonadotropin, or HCG. So now that we have the corpus luteum here, it is not actually going to produce estrogen, even though it's basically a collection of all the left behind granulosa and theca cells. It's not going to make estrogen, it's going to make a different sex hormone called progesterone, which is going to have a slightly different function than the estrogen that we've seen before. But at this point, now that we're producing progesterone, we do restore the classical negative feedback, in which this surge of progesterone concentration actually inhibits the hypothalamus and anterior pituitary from making the earlier hormones in the chain. So, like we said, the ovaries have now ejected the primary oocyte into the peritoneal cavity, so that's the ovulation phase, and once we get into the luteal phase, we start making a whole bunch of progesterone. So after all that's occurred, we need to stop thinking about what's inside the ovary and think about what's happening outside the ovary. So the fallopian tubes in this diagram can be seen to serve as kind of a conduit that connects the ovaries into the uterus, which is where the fertilization is going to happen. So the problem here is that we need that ejected oocyte to migrate into the fallopian tubes and migrate through into the uterus. But the issue here is that oocytes don't have flagella like sperm do, so they can't exactly swim there. So they're basically just kind of going with the current here. Wherever the currents take them, that's where they're going to go. So it's a good thing then that the the luminal side of the fallopian tubes have little beating cilia that actually create a current that will suck that ejected oocyte into the fallopian tube and pull it in. So you've got these little cilia beating back and forth, which creates a current that draws the oocyte into the fallopian tubes. So taking just a very general look here, the uterus itself, we've already mentioned two of the histological layers. The perimetrium we have not talked about, so that's just the outermost layer of epithelial cells, which acts as a shield. The myometrium we have talked before. It's the middle layer of smooth muscle that produces contractions during childbirth. Do you remember what hormone triggers those contractions? I wonder. And then finally, the innermost layer, which is the endometrium, that innermost layer of connective and epithelial tissue, which thickens during the folliculogenesis process and then sheds during menses if fertilization does not occur within the allotted time frame. So this diagram that you see here should be helpful because it actually tracks the concentrations and relative levels of some of these different hormones during our 3 different phases, the follicular phase, ovulation, and the luteal phase, not to mention the menstrual phase if, again, that fertilization does not occur within the 28-day window. So if you look here, we're tracking really 4 different hormones here. Follicle-stimulating hormone in red, luteinizing hormone in blue, estrogen, which is spelled really weird here in yellow. This must be a Dutch or Scandinavian diagram. I don't know who the heck spells estrogen with an O, but to each their own, right? And then progesterone here in green. So probably the easiest thing to look at here would be estrogen. So estrogen, you can see, really starts rising during the follicular phase because those granulosa and theca cells are responding to debatably low levels of luteinizing hormone. So estrogen levels consistently start to rise even as the luteinizing hormone levels start to rise towards the tail end of the follicular phase. And then, as we said, that luteinizing hormone peak is going to be what triggers the ovulation process, right? Okay, so if we look at follicle-stimulating hormone, that's going to really start to rise towards the first part of the follicular phase and then really kind of start to peak about the same time that luteinizing hormone does. And again, it helps to kind of categorize that in the same way because it is one of the things that happens right before ovulation. But let's look at progesterone here. Progesterone stays pretty quiet until right after the ovulation process, after the formation of the corpus luteum, which, as we said, serves as the structure that actually makes the vast majority of the progesterone. So it makes sense that progesterone really starts to peak during the luteal phase and then stays pretty quiet during the other phases. So yeah, I definitely recommend you study this chart pretty closely so you should be able to make sense of all the different events that we've talked about before and make sense of each hormone with respect to one another. It is worth mentioning that in terms of birth control and this diagram here, a lot of different forms of birth control are basically just a big pill of either estrogen or progesterone. So if this pill is taken prior to the follicular phase, meaning before you get to this event right here, you actually activate the negative feedback loop earlier than it's intended, meaning you never see that surge of luteinizing hormone and follicle-stimulating hormone because that estrogen or progesterone pill that you took activated the negative feedback and prevented those surges from occurring, meaning ovulation does not happen. And then finally, if we look at oxytocin and the childbirth process one more time, even though we've looked at it several times, now that we've talked about some of these specific events in the female reproductive system, we can kind of bring everything full circle here. So the cervix, which is the most inferior portion of the uterus that projects into the vagina, this, as we said before, contains those sensory receptors that sense the pressure that the baby about to be born places on the cervix as it starts to descend into the birth canal. So that really starts to get this positive feedback loop rolling in which the cervix is stimulated, which triggers an afferent signal sent through sensory neurons to the brain, specifically the hypothalamus. It will then trigger the release of oxytocin from the posterior pituitary, which then gets into the bloodstream and stimulates the myometrium of the uterus. And you know the story from there. We get contractions, the baby gets pushed along further into the birth canal, we get more pressure being placed on the cervix, and then more oxytocin, more contractions, and the whole thing just repeats ad infinitum, or at least it would if the baby didn't eventually get born, which is what eventually causes this whole loop to be broken. Okay, so that is going to do it for our admittedly brief look into the reproductive system. We could probably teach a whole semester's worth of class on just this topic, but you should feel fairly comfortable in that you've gotten a basic overview. So that should do it for now. I will see you next time for the next video.